Professor Carlos Bustamante, it's a great pleasure to have you here. You are interested in studying how the cell converts chemical energy into mechanical work through highly specialized molecular machines. I'd like to start by asking you, what are molecular machines?
Yes, well, thank you, Maria Celia. It's a pleasure to be here. And yes, the view of the cell suffered a very dramatic change some 20 years ago, 25 years ago, when it became clear that the cell was not just a small bag with a concentrated solutions of proteins and nucleic acids, as was the description that they had of the cell when I was a student. There was no conception that many of the central functions of the cell are carried out by specialized factories that are in charge of transcription or replication or protein synthesis, etc. So this concept, this new concept of factories led to the study of those molecular species that were involved in these factories. And soon after, it was clear that actually they function as molecular machines or molecular motors, to be more precise. Molecular motors because essentially they function as energy transducers between chemical energy and mechanical work, which is often required in order to carry out the function of these molecular machines. For example, when you replicate the DNA by a DNA polymerase, it's not just synthesizing a bond between the 3' end of the growing chain and a new nucleotide that is added, but you have to actually make sure that this process occurs unidirectional and that this process occurs in more or less processive fashion. And for that to happen, you need to spend energy and convert some of the chemical energy that you spend, for example, by breaking the phosphodiester, by releasing a pyrophosphate in the case of DNA synthesis or RNA synthesis in transcription. Part of that energy is used to actually ensure that the machine, which is the enzyme that is doing the catalysis, moves in one direction as opposed to just diffusing randomly. So all these machines actually end up by and large moving as functioning as molecular motors or energy transducers.
Okay. Your laboratory applies new methods of single molecule detection and manipulation.
Yes.
Could you briefly explain these methods?
Yes. So many years ago, we did an experiment that essentially had no purpose other than it was fun to do the experiment, which is to grab a molecule of DNA by the end and pull it. And we asked, how much force do I have to apply in order to extend the molecule of DNA? Like a rubber band, and we were studying the elasticity of DNA molecules. I would be lying if I say that at the time I had an idea that this very fun experiment could eventually be useful to study molecular machines. I had no idea at the time, because we as scientists can never we never see the future better than anybody else. Essentially, we are just working, trying to push the boundaries of the present into the future. But so I didn't know, but it turns out that actually that simple experiment meant that it was possible to mechanically manipulate molecules using femtonewton and piconewton forces, and that it was possible to measure the displacement that was produced by these forces. And out of this idea came out the field of single molecule manipulation in biophysics. And I still remember that actually I went to the Biophysical Society meeting in Baltimore the year that we have done this experiment of extending the molecule of DNA, and I had asked for giving a talk, a presentation, an oral presentation, and they told me, no, you can present a poster if you want, but you cannot present a talk. So okay, so we went there with my students and collaborators and we presented the poster, but pretty soon people started passing the word. And so people would come and say, oh, you should go and look at the poster number 103, 105, 103, you know, and then people would come and all looking around in the poster. At that point, I got the first inkling that actually we were into something interesting, that something that could be potentially useful beyond the fun of doing the experiment. And from there, we moved into using optical tweezers, which is a technique that I have described today, where you use essentially light to trap matter. And light is very convenient because it's very easy to actually use light and manipulate light. And if you can manipulate light through optical tweezers, you can manipulate matter. And if you attach beads to your molecules, you can actually study protein unfolding and protein refolding, and you can study all these molecular machines. You can apply forces, and you know exactly how much force you are applying. You can detect displacement due to the application of forces or due to the movement of a molecular machine, of a molecular motor. So there are many, many ways in which you can actually work, investigate these processes using force or torque as a way to either you apply force or torque to the molecules and you look what happens to the molecules, or you can actually detect the generation of force or torque by biochemical systems, right? By biochemical systems. So it's a very exciting area, it's a new area. And in fact, it's interesting that now you go to the Biophysical Society and between single molecule fluorescence experiment and single molecule manipulation, you have a majority of posters and presentations are about single molecule, right? I mean, so...
And yes, I would like to ask you, what are the advantages of studying single molecules?
Yes, well, the advantages are that actually when you study a process that occurs dynamically in time, and you have molecules, many molecules in solution, what we call bulk studies, or we call them in multiplo studies, many, many molecules, you can never synchronize the molecules to do the work. So you end up actually always averaging over molecules that are dephased, that have lost coherence with themselves. And the averaging is always a loss of information. You end up actually seeing that, for example, when you add urea to your solution of proteins, yes, if you follow with spectroscopically, with a, it's just a spectroscope, you follow the spectrometer, you follow some signal of tryptophan, for example, yes, you will see a decay curve that will occur within 5 minutes, but what you're seeing is an average of what's happening in solution. In solution, there will be molecules that are unfolding right away, others that are would unfold very late, others that are unfolding in the middle, and your average does not represent really what is happening. So averaging is dangerous, doesn't give you information. The solution to the problem is to actually study one molecule at a time, because then the molecule is always in phase with itself. It's not, you are following the molecular trajectory of that particular molecule, and then you study many molecules and you can get the distribution. But you know the kind of phenomena that are happening at the single molecule level, and you don't average those.
Okay. Could you talk a little about your current scientific goals?
My current scientific goals? Yes. Well, we are very interested in continuing study. In my laboratory, we are studying several processes of the central dogma, transcription and translation, and we are planning to continue studying those aspects of the central dogma at the single molecule level. We are also planning to, have become recently interested in the process of condensates of this liquid-liquid phase separation that has become very popular these days, a very exciting area in biophysics. And in fact, we just published a paper in bioarchives where we study the process of liquid-liquid phase separation in nucleosome arrays. And we use cryo-electron tomography, and we were able to actually detect the organization and where each nucleosome is in these globular condensates, which had never been done. This is the first time that somebody has used electron microscopy to actually see the inner structure of one of these droplets, what are called droplets, which are seen inside cells in vivo and in vitro as well. So I'm kind of excited about this development, which is really kind of different from the other things that we do, which are more manipulation and use of force and torque to study molecular machines. But it's all related in a sense, because it is believed that this condensation is a mechanism by which chromatin may go from a state of that is capable of being expressed in terms of transcribed into regions that actually are sequestering this chromatin in the form of heterochromatin, and that this heterochromatin actually is inactive, right? Is silence, is a way of silencing genes. So these studies are new. We just sent the paper for review, and we have put the bioarchive paper just for the people to actually know about that this is the work that has been done, and that with some luck we may get it published.
What are the possible applications of your research?
In terms of practical applications?
Yes.
Well,
yes.
I have one student, for example, that is very interested in understanding a protein that is very important. It's called RNA polymerase, but it's RNA polymerase, not any RNA polymerase, but it's RNA polymerase from Mycobacterium tuberculosis. This RNA polymerase from Mycobacterium tuberculosis is the main target of antibiotics in the fight against tuberculosis. And so what has happened is that in several countries, including India and also Peru and Pakistan, a number of strains have appeared of the Mycobacterium that are totally drug resistant. All the antibiotics that are used today, which are the first line antibiotics against tuberculosis. So it's something dramatic to actually see a young or old, for that matter, person getting the infection, and then in the course, and that nothing that you can add, put in their body helps, and 3 or 4 years later, this person dies because there is no way to fight this. So there is a lot of interest in actually studying, developing new antibiotics, new strategies to actually fight and this molecule, neutralize the RNA polymerase from Mycobacterium tuberculosis. And we are studying Mycobacterium tuberculosis, and we have a collaboration with a group of scientists that are synthesizing new antibiotics. And what is good about our method is that it gives us intimate detail about how is that the antibiotic blocks the enzyme, whether it slows it down or it is actually makes it pause or makes it detach from the DNA or whatever. So we get much, much better information about the mechanism of inhibition than you can get from classical biochemical studies.
Very interesting.
We are hoping that actually this would be something that would become very, very, very useful for the fight against tuberculosis.
Yes. Professor, how do you see the future of of your field, and what are the key challenges?
Yes, well, if I think of my field as being single molecule studies, force spectroscopy, manipulation, I would say that an important development that needs to actually occur is to be able to work under conditions that simulate more and more the interior of the cell.
Mm-hmm.
In most of the studies, all of the studies that we have done are in vitro studies. In singulo, because we are using single molecules, we call it in singulo, but in vitro as well. And but that's not how things happen in the cell. And so what I really think that will be important is that we should begin to actually work, for example, with cells extracts,
Mm-hmm.
and study the systems that we are studying in the presence of the many other components that the cell has, and see, for example, what is the changes that we observe in the presence of those many components. And at the same time, we can, for example, do selective depletion of certain factors. We can immunodeplete the certain factors and see what happens when I take away this factor, or what happens when I take away this other factor from the cell extract. And in this way, begin to bridge the gap between in vitro and in vivo studies.
Mm-hmm.
In vitro is wonderful because you have only very few variables and you control them all. So you can vary one of them while you keep the other constant. In vivo is very rich because that's how it happens in the cell. But the problem is that you don't have control.
Mm-hmm.
And so we need to bridge those two. And I think that if we work with cell extracts under control conditions in which we can deplete selectively certain factors, then we may be able to actually make a bridge for the first time between in vitro and in vivo studies.
Yes, yes, it would be a great advance, I think.
Yes.
What do you enjoy most about science?
All right. Well, my answer is going to surprise you. What I enjoy most about science is actually working with people.
Oh.
Because I happen to believe that there is only one thing that is more interesting than these molecular machines and these molecules.
Mm.
And that thing that is more interesting than molecules is human beings.
Okay.
I really think that actually is a wonderful thing to be able to work with students and associates and discuss and have exchanges where you can see their intellect and their creativity actually, and how they propose experiments and how they discuss among themselves. It is something really wonderful. I think the individual, the scientist itself is a person that is training or has been trained to think scientifically in a scientific manner is a really wonderful experience to actually behold, because the individual is the one that has the creativity, the individual is the one that comes up with the idea, that designs the experiment, that carries out the experiment. So I find that actually working with people, working with other people, with other human beings is really the most interesting aspect of science, in my opinion.
And highly complex also. Highly complex also.
Yes, much more complex than molecules for sure. But,
Yes.
But therein, that complexity lies the mystery of creativity.
Mm-hmm.
So we cannot have one without the other.
Yes.
We cannot have creativity, the intellectual creativity of the scientist, and at the same time actually having this person to be somebody simple,
Yes.
a simple system. We are complex beings, highly complex beings, but in that complexity, therein lies the wonder of the human mind, right?
Yes, sure. Now, Professor, how important it is for you to participate in the 20th IUPAB Congress in Brazil, though virtually?
What is the question?
How important it is for you to participate in this Congress?
Well, I must say that actually, I was born in Peru. I'm a Peruvian and also an American citizen, but I love La Patria Grande, you know, the big country, which is the countries in Latin America, the Hispanoamerica, and Iberoamerica, to be more precise. And I have made a point in my career when I got the possibility at some point in my career to actually extend collaborations with countries like Argentina, Brazil, Chile, Peru, and Mexico. And so I really think that actually is when I got the invitation, my first reaction is immediately when I see that it's going to happen in Brazil, my immediate reaction was to say absolutely yes, because it's Brazil, not because I want to participate specifically in one more conference, but because I love the idea that actually I can help in any way to actually further the research possibilities and capabilities of our countries. I think that actually there is a wonderful human capital in Iberoamerica that many times do not have the opportunities to actually get to the best laboratories in the world. And if that situation is going to change, it's going to be on the basis of training better and better our young scientists in South America, in Central America, and to actually have eventually some of them go back to their countries and potentiate the research. So for me, participating in this meeting, it was a letdown because it was going to happen before, as you know, it was going to happen in 2020.
Yes,
so
yes.
I was prepared for that, but then COVID came and it changed everybody's lives.
Yes.
And so I said, well, I will not be able to go to Iguazu. I think it was going to be in Iguazu, which I don't know.
Yes, it will be.
Iguazu, yeah. But I said, okay, that's the main problem is that now I will not be able to interact with Brazilian scientists and scientists of other countries of the region. But I was happy when they said we are reorganizing it and it's going to be remote and it will happen in October. So, by the way, it was going to be a joint meeting of the Brazilian and Argentinian Society. And in the end, because of the COVID, it couldn't happen. So now the Argentinian Society of Biophysics is having a separate meeting, and they contacted me, and of course, I could not say no. So I will actually participate there as well.
Okay. What's your advice for the young scientists?
Yes. Well, I think that being a scientist is both a privilege and an obligation, in a sense. We are the people that society has put in charge of asking the questions, and that is not an easy job, because being a scientist means, as I have said it in other occasions, to be living permanently in the borderline between our competence and our incompetence. Why? Because the scientist is pushing the boundary of knowledge. And being at the front field of knowledge means that every time you give a step forward, you are in unknown territory, and you are just as incompetent as somebody who is ignorant, ignorant about scientific terms. And if you give a step backward, then you feel comfortable But being a scientist, you are a scientist not to feel comfortable. You are a scientist precisely to live all your life feeling uncomfortable, feeling that you don't understand, feeling that you go back to your house and thinking it doesn't work, the model doesn't work. How is it that it works? Constantly being in that position. See, that's different from a doctor. A medical doctor knows that actually this is what can be done with this particular case and this particular disease. And if the patient dies, well, medicine did all it could, is what we know today. But, and the doctor enters the hospital and comes back at the end of the day out of the hospital feeling comfortable, perfectly comfortable with himself or herself, because that's the way medicine is. It's really an application of knowledge well established. The scientist is creating the knowledge. The scientist is creating the knowledge, and that requires that we accept that we feel incompetent, uncomfortable. And so I often tell my students, do you feel like you are incompetent sometimes? And they almost always say yes, yes. I say, great.
That's good.
You are doing your job. What would bother me or concern me if you tell me, no, I'm perfectly comfortable or perfectly competent. Then probably you're not doing your job.
Very interesting, this vision.
Yeah, and I tell students, just follow your heart. I was supposed to be a physician because my father in particular wanted me to be a physician. I discovered first biochemistry and then biophysics, and I decided that I wanted to be a scientist. And I think that actually you cannot do anything if you don't do it, if you are doing it fighting against yourself. Because there is only one person that can never defeat you, or that you can never defeat yourself. So if you are actually doing science, is a challenge enough so that it helps if your heart is behind it. And my final recommendation to students is do not despair when things are not going well. Many times the fight is uphill, and there will be times where you actually feel that you are not done for this job, that the system is not going to work, that you have lost too much time. Do not despair. Sooner or later, nature will yield its secret, but you need to be persistent. So be persistent.
Thank you very much for joining us, Professor.
Thank you. Thank you for the invitation and the opportunity, Maria.